Hey everyone, this is just a quick fix on what you need to know about apoptosis for today and tomorrow's lessons. So apoptosis is otherwise known as programmed cell death, and it's important to always focus on VCAR's words when you're actually studying one of the dot points in 3-4 biology. So VCAR say that apoptosis is a natural regulatory process of programmed cell death initiated after a cell receives a signal from inside, so the mitochondrial pathway, or from outside the death receptor pathway. A cell resulting in the removal of cells that are no longer needed or that may be a threat to the organism. This is mediated by caspases that cleave specific proteins in the cytoplasm or the nucleus. Please be aware that details of specific cytoplasmic or nuclear proteins are not required. So some keywords from here. You need to know about the intrinsic or the mitochondrial pathway for apoptosis. So that's a signal coming from within the cell that says things have gone wrong here, you need to eliminate me. Or from the extrinsic pathway, otherwise known and described by VCAR as the death receptor pathway. So that's when another cell will notice that things that cell needs to be eliminated, such as, so best example is a T lymphocyte identifying that a cell has been infected by a virus and has viruses within that cell, and therefore the T lymphocyte will bind with the cell and activate the death receptor pathway and cause apoptosis to occur. The other things you must be able to describe is that caspases, and again VCAR have told you this in the dot point, caspases are intracellular enzymes. That's what the A's is for, that cleave specific proteins. So they cut through specific proteins, causing the intracellular proteins to collapse and be broken down, therefore resulting in cell shrinkage, resulting in condensation of the DNA, resulting in apoptotic bodies or blebs to form, as shown in this fantastic image. This is also, so Dauchi used this image in his Edrollo. And therefore those little blebs that form can be engulfed by macrophages, and all the intracellular biomolecules can be recycled. Now, it's important for you to know that not every single cell that dies as a result of apoptosis. The alternative is cell lysis or necrosis, which is a much more untidy, wasteful and potentially damaging way for cells to die. So cell lysis is when the membrane physically ruptures and spills the contents of the cytoplasm out of the cell. What this causes is a lot of mess, a lot of wastage, because the organelles and the biological molecules are lost out of the cell and can't be recycled as effectively. Alternatively, well, in addition to that, it causes an inflammation response. So if you think about inflammation as pain and discomfort, then, oh, close that message. Then every time a cell is eliminated from an organism, that if pain and discomfort occurred and a large-scale immune response was initiated, then that wouldn't be particularly efficient, and the temperature would be increased, and the usual things that you see with inflammation, such as swelling and bruising and all sorts of different immune cells attracted to the site, is a very wasteful way to do it. Okay, so cell lysis isn't as tidy, isn't as organized, and is totally avoidable by apoptosis. Just going back to cell lysis, the things that cause cell lysis, and it does occur, is physical damage to the cells. So if the cell is physically the membrane's broken beyond the point where the membrane can reseal, then that can cause lysis, and also things like swelling in response to a hyperosmotic environment and water rushing in, causing pressure on the membrane. That can also cause cell lysis. So it does happen, but apoptosis is a much more regulated and controlled way to remove cells from an organism in a tidy way without the inflammation. And those cells break down into apoptotic bodies or blebs, which can then be engulfed by macrophages and recycled. Some reasons for apoptosis. So we remove cells that have damaged DNA. Okay, so DNA damage is a risk to an organism, and therefore those cells need to be eliminated. damaged membranes, infection with a virus. That's a classic extrinsic trigger to the extrinsic pathway. Things like, so these things here we might call tissue sculpting. So during fetal development, there's connections between the fingers, and those connections break down due to apoptosis during the development of a fetus, and that we tend to call tissue sculpting. When the embryo starts to take shape and the fetus begins to become more defined, the appendages become more defined, that's absolutely essential, and that process is apoptosis that occurs, that actually causes the fused or the tissue between fingers to break down. Shedding of the uterus lining, intestinal cells being replaced, all of these things, bone cells actually undergoing apoptosis due to aging after menopause, many reasons for apoptosis to occur, and essential reasons. One of the biggest ones is to keep the cell numbers in check. So organisms don't want to accumulate more and more cells over a lifetime. It's really important that as new cells are formed, old cells are eliminated to avoid damaged cells persisting within the organism. So the two pathways that you're asked to know about from VCAR's perspective are the extrinsic pathway and the intrinsic pathway. Now it's important to know that when we trigger the extrinsic pathway, that will also cause the intrinsic pathway to activate as well. Okay, so the intrinsic pathway is often cited as a response to DNA damage within the cell, biochemical stresses such as low oxygen or metabolic stresses. And what happens is the mitochondria start to release small molecules, and so the mitochondria becomes perforated, and cytochrome C will start to leak out from the mitochondria. And this initiates an intracellular cascade, which you'll remember from signal transduction. And eventually we find that a whole series of caspase molecules or enzymes are activated. Now some of these are intermediate and don't actually have an effect on proteins, don't break down the proteins themselves. What they do is go on and cause or activate what we call effector caspases, which do act as the digestive enzymes that actually break down the protein and cause the cell to then go on and collapse and undergo the cellular process of apoptosis. You don't need, as VCAR say, you don't need to know all of the proteins involved. One of the good ones I like to remember is the apoptosome, because I think it's a really nice word. It's a great example of a quaternary protein. It's quite complex and quite unusual in its structure, but this is one of the intermediate molecules that's active, well, it's constructed and then causes the activation of caspases, which eventually result in the breakdown of proteins within the cell. So it's really important to keep in mind that the extrinsic pathway is activated by a death ligand binding to a death receptor, as we've got here. And once that happens, a whole series of caspases are activated, eventually leading to apoptosis occurring as these effector caspases, caspase 3 and caspase 7, both here and in the intrinsic pathway, once they're activated, they will actually go and break down the proteins within the cell, remembering that proteins have multiple different roles within the cell, holding that structure through the cytoskeleton, which are like the tent poles of the cell holding it up. As soon as they're broken, the cell will start to collapse. Also, obviously, all the proteins within the mitochondria, within the nucleus, within the membranes, within the organelles, they'll all start to be degraded by these effector caspases and broken down. And this is what causes the cell to then go on and form blebs, which can be consumed by the macrophages and recycled. So some key things for you to remember are that the extrinsic pathway is often initiated by immune cells such as T lymphocytes, which will identify an abnormal molecular pattern on the outside of an infected cell and initiate apoptosis by binding the death ligand to the death receptor. And the intrinsic pathway is often triggered by biochemical stresses, DNA damage, and potentially the removal of growth factors that are there to keep the cell dividing and keep the cell healthy. As soon as they're removed, then apoptosis can initiate. Critical for you to see that these two diagrams come together and the response is the same, the cellular response is the same, the breakdown of those proteins by effector caspases and the collapse of the cell. And this is a series of diagrams that I want you to be able to draw. So you need to be able to show that once apoptosis is initiated, the cell will start to collapse due to the breakdown of those proteins. Blebs will start to form and membranes surround those blebs, and eventually forming the nucleus. The chromatin will become condensed. The organelles will become damaged. Sometimes, not all of them necessarily. You might see intact organelles within these blebs, and then eventually small tiny versions of cells with damaged DNA and damaged organelles will be formed and release cellular signals themselves, which will attract large phagocytes such as the macrophage, which will engulf the apoptotic bodies and recycle all of the biomolecules inside there. So it's a very efficient, very tidy process. That's basically it in a nutshell. So I'd like to make sure that you maybe play this video once, twice, 3 times and extract all the keywords from it. Make sure that you can describe the intracellular or intrinsic process and the extrinsic process. Tell me when the two come together and what the common factors which is the cellular response is in both of those, and be able to draw at a cellular level what's happening during apoptosis. Well done.